Hello everyone. Today, the title I want to present is CamoTSS, Analysis of Alternative Transcription Star Site for Cellular Phenotypes and Regulatory Patterns from 5' iCRNA-seq data. And I will give the introduction from this aspect. First, I will give a brief introduction on the CamoTSS pipeline and the performance of CamoTSS. And then we will use 3 data sets to prove the application of CamoTSS. This is a flowchart of 5' iCRNA-seq from 10x genomics. As we all know, RIS1 always includes UMI and cell barcode. However, sometimes we can find that RIS1 also includes the extra sequence. We think this extra sequence may be the ideal material to detect the transcription star site because it didn't be fragmented. So based on this, we developed the CamoTSS. This is a pipeline of CamoTSS, which includes 3 parts: clustering, filtering, and annotation. Here we use a hierarchical clustering method to do the cluster for the position 0 of RIS1 aligner. And then we mainly use a classifier to filter the false positive. And finally, we use a Hungarian algorithm to do the annotation. This is the detail of classifier. The attack-seq peak was used to distinguish the positive and negative samples, and then around 10,000 samples was fed to our model. For the logistic regression model, we extract 4 cluster features, including the unencoded Z percentage, standard deviation, and summing count and all counts in one clusters. And for the CNN model, it just has a very simple architectures, including the convolutional layers and max polling layers. So here we use the IPSC and DMFB combinational dataset to pre-train our classifier, and then we embed this classifier to our CamoTSS. So this is the performance of classifier. As you can see, no matter the class features, sequence features, and the combination features, they all have a good performance. Because this is a pre-trained model, so we want to prove the generalization of the cluster of our model. So for the cluster model, we use the IPSC to predict the DMFB. And we also use the pre-trained model to predict the PBMC. We found that they all have a good performance. For the sequence model, we use the CTCF knockout model to predict the sequence, which only have the binding site of CTCF. And we also use the E2F6 knockout models to predict the sequence, which only have the E2F6 binding site. They all have a reasonable performance. And then we try to identify the important features during the prediction. We found that unencoded Z percentage and the DNA sequence from the CNN model are the most important features. The unencoded Z percentage that can symbol the if this mRNA is intact mRNA, then it maybe have the unencoded gene. So here we use the histone signal and RNA polymerase II signal to prove the reliability of the TSS detected with bias. The histone signal and the polymerase II signal of total TSS and the TSS located at the exon and the intron, and also include the negative labels, all have signal as we expected. There are two examples. The red line are the TSS detected by us, and you can see they all have the support from the histone signal and the ataxic signal. And especially we also detect the novel TSS, which is not annotated in the CTF file. But here it also has the histone signal support and ataxic signal support. Then we use the 15 organ dataset to prove that CamoTSS can help to identify cell type. And firstly, we calculate the Pearson's correlation between the TSS expression of different cell types from 15 organs. We found that compared with organ, TSS is more cell type specific. So for each organs, we can get two profiles. One is TSS profiles, another is gene expression profiles. These are the top significant markers from the TSS profiles and the gene expression profiles. And you can see here are still some unique TSS markers which can help to identify cell type. These are two examples of TSS markers. So next we compare the cluster difference between the gene expression profile and the TSS profile. We found that in muscle, in the T cell, they have different clusters. We suspect that the difference is due to that TSS profile is more similar with the regulation profile. So here we use the technique to calculate the AUC cell score of regular in each cell, and then use that matrix to predict the T cell clusters. We found that compared with the RNA labels, TSS labels is easier to predict. When we use unsupervised methods, we can also get the same conclusion. In addition, we also found that alternative transcription start set can help to cell type identification that masked at the gene level. Next, we use nasopharyngeal carcinoma dataset to detect the altered TSS usage in cancer. This is a UMAP based on the gene level expression, which was annotated with the cell types and the disease status. Here we use a BRID to detect the differential TSS between the NPC and NRH in different cell types. The LMS1 is a very important gene in cancer. It includes 3 TSS, and we can see in T cell, the proportion of minor TSS upregulated in the NPC. However, in the myeloid cell, the proportion of minor TSS decreased. This evidence shows the complexity of TSS regulation. To review the underlying mechanism, we use a FEMO to calculate the binding site frequency of TF from the Jasper dataset for the TSS detected in the last step, and we found that the TF prefer to bind with the NIH. In addition, some TF in the NF-kappa B family would like to combine with NPC, which is consistent with the literature. In addition, we also observe the expression of this significant TF, and we calculate the proportion of expressed cell of this significant TF. We found that they have the consistent trend with the scatter plot shown in the last slide. In the last few data sets, we all look at the clusters, TSS clusters. However, we also want to check the narrow shift within one cluster. So here, this is one cluster in the last step, and we use the window slide method to remove the noise, and then use the count and function to at the threshold to remove the noise and to detect the CTSS, that is cage TSS, that is at single cell resolution. Here we use a human thymic development data set, which includes 3 time points to check the altered CTSS during different time points. So here we also use a bridge to detect the differential CTSS in different time points in different cell types. As you can see, there are 2 examples. This is a KDN1 cell, KDN1 gene, and they have a shift from the week 11 to the month 13. From here to from this TSS to this TSS, and the distance is very close. To summarize, the CamoTSS is a data-driven method, and it embeds a classifier to accurately detect the TSS. It can also facilitate the cell type identification. In addition, we can also observe the differential TSS usage in cancer dataset, which is related to the transcription factors regulation. We also observe the narrow shift TSS within one cluster in the development dataset. These are our preprint manuscript and our software. My poster number is 341. Finally, I thank the instruction of my supervisor, Dr. Ruiyan Hou, and the help from my lab mates, especially Qiaoshan, Weizhong, and Xianjie. And I also thank the conference organization giving me the travel fellowship. Finally, thank you very much for your attention.
Question for you. I'll start with one. So you show that when you look for explanation in terms of regulation for the changes, you had a bunch of hits for various specific, transcription factors, and you found some multis there, right?
Yeah.
So I'm curious, if you take those into account, how much of the variations in the changes that you observe can be explained by those bindings? How much of the variation in the data in terms of the start sites? Have you looked at this? Did you try to quantify it?
Yeah. We do the quantify. We count them, and in addition, we also do the permutation to prove that this trend is significant. So, do you mean that we count the binding site frequency?
No, I was wondering how much of the variations are explained by the various TS that you're finding, but we can talk about it later. That's okay. So I had another question which has to do with, you said you have this embedded classifier. The classifier looked to perform extremely well. But it seems like, what you're getting is very clean, or am I missing something? And when you showed the features, it was mostly very few features that actually enabled you to.
This for features.
Right.
The unencoded Z percentage is very important because here you can see this is 5' process, and this in the intact RNA always have the cap in the initial location, and the cap is ECCB, and so we calculate the percentage of this unencoded G. If this rate is intact, then it may include the include this DGMP, and then we calculate the percentage. So if it is intact, then we can observe the transcription stem cell. That is a true transcription stem cell. I don't.
Thank you for the help. And the correct TSS is very important, for example, for CRISPR-IA screening or stuff like this. So it's very important to know exactly what the TSS in particular is on there. When you compare your CamoTSS performance versus, let's say, annotation on Phantom 5 database, stuff like this, what is the percentage of overlapping TSS you observe?
Can you speak again? I don't understand that.
So if you compare your TSS, you identify, for example, in T cells, and you compare your TSS, particular genes in TSS that's already existing in Phantom 5 database. You know, the Phantom 5 database is very famous database for the TSS, and you can see that they identify TSS based on the cage. And so if you compare the existing annotations in Phantom 5, for example, with your CamoTSS, what is the number of overlapping TSSs?
So you want us to compare the TSS with the cage dataset? Do you mean that?
Yes.
Here we don't. Actually, we tried some. Here we don't use the cage dataset because it didn't. We don't find the single cell level. It just has a bulk.
Maybe we can continue it later because we need to move to the next topic, okay?
Okay.
Okay, let's thank